Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 6th August 2020. Our memory helps us in understanding and reflecting upon what we have studied so far. If we analyze, we have been talking about the codominance of blood groups, wherein all the genes, all the alleles contribute equally for the expression of a phenotype. Like AB blood group, both IA as well as IB should contribute for the expression of blood group AB. So this is possible because of linkage and recombination. What is that we were talking about? Linkage. And we said that linkage is the genes that are present on a chromosome. If they are present in very close proximity during the recombination of genes, recombination of chromosomes, all will go together. But if there is enough space between the genes, between the alleles, then during recombination the genes get separated or segregated, law of segregation. So this linkage and recombination will decide what would be the parental combination that will form in F2 generation, as well as what will be the new combinations that will be formed in F2 generation. So linkage and recombination play a crucial role in the formation of varieties of children, or varieties of what we call offsprings. So here, as we discussed in our previous session, Morgan carried dihybrid cross in Drosophila to study the genes that are sex-linked, and he crossed yellow-bodied and white-eyed. See here, two characters he has taken: yellow-bodied and white-eyed females with brown-bodied and red-eyed males, and the F1 generation progeny is formed. The two genes did not segregate independently. What we said, law of independent assortment says that every gene, every chromosome will assert itself independently. But here we are seeing a deviation. The two genes did not segregate independently of each other, and the F2 ratio deviated from the original 9:3:3:1. We said that F2 will be always 9:3:3:1, but there is a deviation from this. So the genes present on X chromosome and two genes in a dihybrid cross situated on same chromosome, that is parental gene, combination is much higher than non-parental. So new ones are more, the combination, than non-parental. This is due to physical association or physical proximity or linkage of two genes on chromosome. This effect is called linkage. Generation of non-parental recombination or parental combination is due to recombination of genes combine. They combine in a different way. So Morgan found genes are grouped in same chromosome, and some genes are tightly linked. Why they are tightly linked? Because of the proximity. They are closely associated. So when they are closely associated, naturally they do not get separated so easily. When they do not get easily separated, the formation of new combination or recombination will be obviously less. When genes are present in different chromosomes, the genes are present on different chromosomes. Let's say one gene present on X chromosome, another gene is present on Y chromosome, then the recombination is higher because genes are present on separate chromosomes. And during the law of segregation, law of independent assortment, the chromosomes will separately go. As chromosomes move separately, the genes also will go. So naturally new things, new combinations will be formed highly. So higher recombination. Example, genes for white and yellow, that is white-eyed and yellowing, tightly linked. How much? To the tune of 1.3% tightly linked. Both the genes for white and genes for yellow. Recombination, while genes for white and miniature wings are 37.2. This is because of not tightly linking together white and miniature wings. So white-eyed and miniature wings is a new combination. 37.2% of new recombination. So student Alfred Stewart used frequency of recombination between gene pairs on chromosome as a measure of the distance between genes. That's what we have been discussing. On a chromosome, single chromosome, if two genes responsible for one character or different characters are present in close proximity, then recombination is not possible. Whereas if the genes are present on the same chromosome but have distance, then during meiosis, particularly in prophase 1, where there is crossing over, when crossing over takes place, the portion of chromatids get cut or get exchanged. As a result, hybrid chromosomes will be formed. The genes get exchanged. So when the genes are sufficiently placed at a distance, new combinations are formed. When the genes are closely attached on a chromosome, then formation of parental combination is more, because it is not possible for the two genes to get separated. Whereas if the genes are present on different chromosomes, let us say gene A is present on X chromosome, gene B is present on Y chromosome. So combination of A and B is very rare. So they are separately present, so recombination may take place. But if on X chromosome, let us say gene A, gene B are present with a minimum distance, then it is very difficult to separate gene A and gene B. So what he has done, this Stewart, he has mapped the gene position on a chromosome. That is the great work he has done. And we are taking an example here. In class A, that is we are talking about the Drosophila melanogaster, fruit fly. So 0 with the plus is female, 0 with arrow mark is a male. So in that, the yellow wings, white-eyed, pair of chromosomes, one dihybrid cross we are taking. And this Y and W, that is again, whenever there is a plus, that is dominant gene. Y plus, W plus is dominant over Y, simply Y and simply W. So the combination, parental combination, we have taken two chromosomes with having genes Y for yellow, W for white, and here wild type we have taken. And when we cross, you can see there is exchange in F1 generation. That is YW, one chromosome has come, and law of independent assortment, and the purple color Y and W are what we call bluish color. And here in another product, F1 generation, you got that white and yellow, and then blue one we have got. That is X and Y. And in F2 generation, when F1 generations have intercourse, the F2 products are formed, and in the F2 you can see the parental combination is to the tune of 98.7%. What does it indicate? That genes are present in close proximity. As you are seeing, what is the distance between Y and W? Please see on the yellow chromosome, Y and W, very narrow. Similarly, Y plus and W plus, you can see very narrow it is. The distance, you focus on the distance, it is not even 1 inch. So, as I said, when the genes are in close proximity, formation of recombinants are very less. That's why it is only 1.3%. Whereas when the genes are closely associated, as we are seeing, look at that parental type, 98.7% because law of segregation is not working out here because of the close proximity. You can see in F2 generation that there is a new combinates, no doubt, that is there is a exchange of crossing over. Also you can see in that yellow chromosome a little bit of Y has come, Y plus has come. So the combinations are Y plus W and then of course white type. So you got a wild type, white one, yellow white, yellow, and then wild type, white one, yellow white and yellow. In that if you see the parental combination, what is yellow white and wild type 1, wild type 2 and yellow white. So parental combination is becoming more, that's why it is to the tune of 98.7%. And what about the other side in cross B? Again this white and miniature is taken and wild type. Again I want you to focus on the distance between the genes W and M. See the distance? Compare this distance with Y and W in cross A. Naturally you are able to observe a lot of distance. So when the distance between the genes is very large, naturally law of segregation takes place, law of independent assortment takes place. As a result, you are getting in F1 generation a wild type and white miniature. What is that we have taken? White miniature, W and M. And on the other side we have taken W plus M plus. So you are getting a wild type and white miniature. And the intercourse in F1 generation will lead to the formation of F2. And here again, I want you to focus on the percentage of parental type. See in cross A, the parental type is 98.7%, whereas in cross B, it is only 62.8%. Why this much less? The reason is the distance between the genes. And when the distance is sufficiently enough, space is there, then the law of recombination will take place. As a result, recombination types are to the tune of 37.2%. What about in cross A? It is just 1.3%, whereas in cross B it is 37.2%. So you can see the new things that are formed. What is the new thing? It is see W plus and M. W and M plus. So you can see here parental combination. Compare the parental combination. What is the parental combination? It is white and miniature, wild type. Whereas here you can see all new combinations have been formed. And how many new combinations? It is to the tune of 37.2%. So this is how linkage and recombination will influence on the phenotype of F2 generations. This is what is discovered by Morgan. So as you are seeing, linkage results of two dihybrid crosses conducted by Morgan. Cross A shows, as you are seeing, cross A, this is cross A, shows the crossing between gene Y and W. Cross B shows crossing between gene W and M. Here dominant wild type alleles are represented with the plus sign in superscript, as I said just now. The strength of linkage between Y and W is higher than W and M. Why? Why strength between Y and W is more? Because of the narrow gap. And why in W and M the strength is weak? Because the in between W and M genes there is a lot of gap. That's why recombination is possible. So linkage is physical association of genes on a chromosome. As we said, how much associated? They are closely associated. So if they are present in group also, sometimes the genes will be present on a particular locus. Locus is the place or position on a chromosome. So on a particular locus or loci, if maximum genes are associated or present, then linkage will be very, very strong in that. Recombination, on the other hand, the generation of non-parental gene combinations. It's not parental, new one, new combinations. And this is possible when linkage is weak. So when linkage is weak, recombination becomes strong. It occurs in crossing over of chromosomes, as I said, during meiosis, particularly prophase 1. And sometimes mutations also take place. What is that mutation? Anything that happens suddenly in an organism is called mutation. So phenotypic variation occurs due to sudden change. It is in gene. The change may be in gene or DNA sequence. So the organism that undergoes mutation is called mutant. Phenomena which result in alternation of DNA, change in DNA sequence, result in change in genotype as well as phenotype. So the mutation can be because of deletion, loss, or insertion or duplication of a segment of DNA results in alternation in chromosomes. So it may be abnormalities or aberrations. So then we say if there is part of chromosome is lost or change in the structure, then we say chromosomal aberrations. And gene mutations, the mutation takes place due to change in a single base pair of DNA. Like for example, I said if you take one base pair of nitrogenous bases, I said always one purine combines with one pyrimidine, like adenine, thymine, or guanine, cytosine. If there is any mutation, it's called point mutation. One mutation like G and C. If C is missing, then that results in a lot of changes in the phenotype. And that changes in the phenotype is because of lack of just one nitrogenous pair called cytosine C. So then we call it as point mutation. Example, sickle cell anemia. And frame shift mutations, either deletion or insertion of base pairs of DNA. Either we will remove the pairs, one purine and one pyrimidine, or insert one purine and pyrimidine, nitrogen base pairs. Then we call it as frame shift mutations. So because of these mutations also, there can be alternation in the phenotype in the organisms.